Food, drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Barros, thank you for joining me.
You're welcome. Thank you.
Where are you calling in from today?
This is Valdivia, a city which is about 100 kilometers south of Santiago, the capital of Chile. This is the Lake District region.
Ah, okay. And can you tell everyone a little bit about who you are and what your lab studies and what you do?
Well, I lead a group of neurobiologists interested in brain metabolism. So we started looking at glucose transporters many years ago, and we've been going deeper and deeper into tissues and cells. And nowadays, our main subject of interest is mitochondria. Mm-hmm. So we study mice mostly, cultures and slices and some experiments in vivo, and try to figure out metabolism, which is something we used to think was solved, but not so, it seems.
Yeah, I mean, in the context of the brain and thinking about how the brain works and how information processing and neural networks and circuits works and does all the interesting things that, people interested in neuroscience are actually interested in, like memory, like learning, like cognition. At least in my formal education, we really sort of siloed metabolism away. It was a separate field of study. Yes, of course, neurons are cells and they have to have energy. They have to make ATP and they have mitochondria and all these things. But you just sort of take it as a given that, okay, all of the energy, if everything's working and you're not, having a stroke or sick or injured or whatever, the blood will bring you all of the energy and the nutrients you need. And then the neurons will do their thing. And we can think about all of the interesting neuroscience stuff. And we sort of just took it as a given that, okay, the metabolism stuff is just there. And that's a separate field of study. And we may or may not be interested in it as neuroscientists. But I think as we're going to discuss, the lines there get really blurry really quickly because information processing is tied up in metabolic signaling and metabolism beyond just the fact that all of the information stuff requires energy.
Correct. You put it very clearly. This maybe it's to do with the computer metaphor of the brain. We have our computers, we plug them or we don't. There's no shades in between, and we don't care if what happens with energy within the computer as long as it has it. But clearly, this is not the way it works in brains because first of all, the energy is not a given, has been in shortage throughout our development as a species, and before that, there are many clues as these short times are very difficult in terms of energy procurement. And now, during development, energy is super important to design the circuits. So the brain uses a lot of energy to do the stuff. We know this is excitatory neurotransmission. Most of the energy is used by neurons and postsynaptic neurons. And then metabolism produces energy on demand. This used to be the classic paradigm. And most of the time, this was produced okay at the right rate, and when this went wrong, you had disease. But something that we have been thinking for a while that doesn't fit with that simple picture is that many neurotransmitters are metabolites, which are very close or are part of the core, the energy core of the cell. Starting with glutamate, glutamate is amazing how we are used to glutamate being 90% of the synapses of the brain are glutamatergic. But glutamate is an amino acid and is central to the core. So every time something goes wrong or in any direction, glutamate levels will change. And here is like the electricity through your circuits of your computer are affecting your software, right? Which is a different way to think and escaping a little bit from the computer metaphor, which is actually weak in many other senses.
Yeah, and so when we think about the brain in this computer with this computer metaphor that you commonly hear about it, the brain is, we think about it as a computational organ. Neurons and circuits are computing things. They're likened to computers in many ways, but you've already hinted at like a major fundamental difference between computers and brains, and that has to do with energy itself and how efficiently it's used. Can you just say a little bit more there in terms about how brains are different from computers in terms of how efficiently they actually use their energy?
Yes, of course. We don't really know how to measure the computing capacity of the brain. This is something hard. But people have tried to use, say, parameters like computations per second, like flops. And if you compare any ordinary computer with the brain that does the same kind of order of magnitude computing processing, the human brain would be about a million to a billion times more efficient than any computer. So to do the things we are doing now, talking and processing imaging, processing language, a computer that does the same will spend a million times more energy and in terms of watts. So this is interesting
this is maybe why all of these fancy large language models, when people hear about how this works, they hear about all these giant data centers that have all of these GPUs running. It's because we need a lot of energy to power this thing.
That's right. You do your search in Google, and that will use a lot of energy in terms of heat released. And somehow our brains, even though they are not doing the same thing, they're doing similar things much cheaper. So this is now a practical question. So if you want to develop a cheaper computer, a more environmentally friendly computer, you would like to understand how the brain does it. And something very important for the brain also is that you cannot replace the circuits, right? Your neuron is gone for good.
Yeah.
So, and this happens throughout your entire lifespan, say 80, 90 years of using the same neuron once and again and again and again. And the computers and the neuron is still there, even though there have been periods in which that neuron is using a lot of energy beyond the possibilities of that particular neuron are taking resources from the neighbors. So there is a lot of there's an ecosystem of energy management which we don't know the rules of very
Right.
much.
Right. And this is a bit of a tangent, I think, from what we'll focus on, but I think a lot of times in cognitive science, broadly speaking, neuroscience, computer science, all of these things, we often think about the computational side and the computer science side and the engineering side as a way to try and understand the brain and the mind side. But things can also go in the other direction. I think what you're starting to get at too is if we understand more about metabolism and energy efficiency in biological systems like brains, it could actually inform us into the design of new computational hardware and software systems that are actually more energy efficient.
Yeah, and this beautiful book by George Sarkanakis that wrote a few years ago that puts your question into maybe into the right context. In say 2000 years ago, people used to think about the brain in terms of clay pottery, because this is what they had in mind. So later on came in during Roman times the metaphor of hydraulics, because the Romans were very good at doing pipes and things like that. Then people thought the brain had to do with the liquids moving from one side to the other. And then later on, the machinery of Descartes, pieces. And then the electricity thing. So every generation or few generations, they use whatever they have in mind, the outside world, the scientific world, and create the new metaphor that would explain the brain. And the metaphor today is a computer metaphor, which just by extrapolating the previous silly metaphors, we would think that this is a silly metaphor we are living in. So in the title of this paper, we talk about information processing, which is a term that comes from computing. But we don't really do that. The brains, the human brains, the mammalian brains don't do information processing in the sense that is used for computing. So the challenge here now is to develop the new metaphor that will replace the computer, because it's really failing as a metaphor, and it's not really giving us the deliverables that we would expect in terms of understanding. So, and this divide between metabolism and information processing is part of the change of the new metaphors. In the new metaphor, we would need to incorporate energy metabolism, the limitations, the possibilities as a part of the metaphor. And now it's not, because of the computing metaphor. So this is exciting times, because this piece of work we have produced, we think is part of the new metaphor that is going to be better than the current one.
And one of the concepts that I want to bring to mind for people that you talk about in this paper that I looked at is this idea of the core of a neuron, or the core metabolic machinery of a cell, versus the things that are involved in intercellular signaling, the things that make a particular type of cell more unique in its function compared to other cells. And so basically what we're saying here is, on the one hand, all of the cells of our body are cells, so they have things in common. They all have to have a cytoskeleton that gives them a shape. They all have to have energy metabolism, right? They've got mitochondria. They can transform glucose and fatty acids into ATP, the energy currency of cells. But at some point, they become different. Neurons are different from skin cells, or different from liver cells. They can do different things. So on the one side, they have this common core of cell stuff, including the core metabolic machinery that they have in common. But on the other hand, they become differentiated, and they can do things functionally and have things structurally that the other cells don't have. And this is a sort of convenient dichotomy to think in. So when we think about the core metabolism, the core machinery of that, the apparatus that is involved in this core metabolic function that is common to all cells, what are some of the major pieces there in terms of the machinery? I mean, we can just sort of take it as a given, I guess, because I've done a lot of content on this before. They're all going to have the ability to do glycolysis. They're all going to have mitochondria doing oxidative phosphorylation. That stuff is going to be in all cells to some extent or another. Some might have more mitochondria than others, and so on. But what are some of the core pieces there, and the core metabolites that are part of this core that's common to most or all cells?
So the brain is quite simple, because the brain is fuel monodependent. It only metabolites glucose for practical purposes. So 95% of the energy of your brain comes from glucose. This is not like other organs, like heart or liver or kidneys. They take more stuff. They take amino acids, they take fatty acids. So somehow, and for a reason we don't know, the brain decided to be solely dependent on glucose. So the beginning is simple. Glucose goes into the brain, and the end point is very simple too. It's CO2 coming out.
Yeah.
So it gets split into several stuff. There are about 30 compounds along the way from glucose to CO2. And to do that splitting is using oxygen. And the energy that is released is harnessed as ATP, and the ATP is useful for all the wonderful things that the signaling, basically, the restoration of ion gradients and so on. So this is the core that is present in a bacteria, in a mycoplasma, in a yeast, in a liver, wherever. Now, as you well said, we have these other metabolites which are much more specialized, which are away from the core. So acetylcholine, serotonin, noradrenaline, they are separated from the core by several out of equilibrium reactions. So they are regulated.
Mm-hmm. So when you say they're separated from the core, you mean that there are multiple steps, multiple sort of energetic requiring steps that take you from the core to these more functionally specific compounds that are a little bit more specialized in how they're used.
That's right. That's right. The precise way to say it is thermodynamically shielded. So the core, say if you want to go to noradrenaline, you have 2 or 3 reactions and you have to pack noradrenaline in vesicles or whatever. So if there are changes in the core because you're hypoxic, that will not necessarily affect noradrenaline deposits. So in that sense, we have a division between the old ancient part of the cell and the more sophisticated new one. And most neurobiologists today will think that makes a lot of sense because you have to be a neuron. I mean, you have to be a cell because this is how developed, but you become less and less of a cell and you become a part of the circuit, right? This is the differentiation.
Right.
The question comes when you see that many of these core metabolites are now heavily involved in the signaling, like glutamate, like lactate, like beta-hydroxybutyrate, the ketone body. And you say, oh, well, maybe this is a trace left from evolution in the making. That could be, right? But then you see Drosophila, and most of the synapses of the central nervous system in Drosophila are acetylcholine. And in humans, it's not at all. It's glutamate. Then you say, well, you could say maybe Drosophila are more evolved, so to speak.
Yeah.
And then comes the discussion, because you need to change your frame of mind, right?
Right.
If you escape from the computer analogy, then you say, oh, maybe this is integral part of the design, that the core is part of the signaling for some good reason. And the some good reason that we came across with is neuroprotection.
Yeah.
Most of these core metabolites are neuroprotective.
I see. Most of the core metabolites are neuroprotective. Maybe we'll circle back to that. Another thing I want to give people here conceptually is, okay, so we talk about the core metabolism that's going to be common to all cells because all cells need a metabolism. They all need to do these energy transformations to do whatever it is they're doing, whether or not they're neurons or some other cell. But then there's signaling between cells. All cells need to talk to other cells around them, or maybe even far away from them. Neurons would be a great example, right? We've got a lot of neurons. A lot of them reach pretty far away and signal. They literally release chemicals. They signal in different ways to other cells. And so I guess classically you would have thought like, okay, the core metabolic machinery, this is an intracellular thing. It's inside of this cell, and this cell will have its own metabolic machinery that uses just to make its own fuel to power all of these intercellular signaling molecules that it will have to do its job as a neuron, say, and then use those to communicate and do stuff between cells. And these two things, you could have thought, these two things are going to be completely separate and segregated. They're going to be thermodynamically insulated from each other. But what you're starting to get to already is that a lot of the components of that core metabolism, that's the pieces that are common to all cell types, they actually get used in signaling between cells, even for things like neurons. So, as you stated, glutamate is a pretty what we think of as a simple or primitive molecule, maybe, but it's used as an intracellular signal by neurons. So it's not just that the signaling and the information processing and communication stuff relies on these functionally specialized or special cell type specific molecules. They do use some of those molecules, but they also use molecules that are the core metabolites of the stuff that's sort of in every cell.
Yeah, correct. And it is some of them, right? It's not all of them. So
right.
From glucose gets into the cell and gets trapped as a phosphate, glucose phosphate. This negative charge traps the glucose in the cell. And then you have several phosphorylated compounds until you have one that is not, which is pyruvate. And pyruvate is connected with lactate, which amplifies whatever change in pyruvate concentration there is. But lactate can move freely between cells through very abundant monocarboxylate transporters. So there you are. You have a way in which the core becomes leaky on purpose, because this protein, this molecule lactate, doesn't have a phosphate. And lactate moves out of the cell, moves between cells, moves away. When you do exercise, your lactate levels can go up to, say, 10 millimolar. And we know now that lactate enters the brain and changes the lactylation state of mitochondrial proteins. So your muscle is signaling your brain in real time as you do the exercise.
Right.
So, and the meaning of that signal, we don't know yet, because this is something very new. Lactylation was discovered 2 or 3 years ago. And lactate is one of them. Beta-hydroxybutyrate is another one. Succinate is another one. These are part of the metabolic core, which are used to inform the environment of that cell that something is different, something is amiss. There's something to have a response about. And in the context of the brain, we think that is the purpose of this signaling, or part of the important purpose of the signaling, is to restrain the activity of the network, because the network is a tyrant. Imagine you're a poor neuron that has to receive orders from that network all your life. What if those orders are too much for you?
Right.
Then this would be a way from those cells to say, stop, enough is enough, control yourself, network, and stop your firing. This would be a simple way to put it, but of course it's more sophisticated than that.
Yeah, and literally, if a neuron is too excited or receives too many inputs too quickly, it can literally kill the cell through excitotoxicity. You can get epileptic behavior and all of these things. So there must be these sort of provisioning mechanisms at different levels to make sure that energy is allocated and not too concentrated in any given part of the body or the brain.
Yeah, there are strategies that we knew from previous work by physiologists. So sparse coding is one of those strategies. There's a beautiful paper a few years ago that they showed that if during development the fetus is food restricted, they will develop circuits which are cheaper in terms of their AMPA receptor conductivity. So we know not only during ontogeny, but not only on phylogeny, but also ontogeny, there are ways to adapt to network demands. And this core metabolite might be part of that strategy in real time, so second to second. So imagine you're using your auditory cortex, and some neurons are firing at 300 hertz because you're listening to what I say. And those neurons, they didn't know that was going to happen a few minutes ago. So they are giving this task without warning. So how do they do to survive for 80 years with these surprises that the environment gives us every day? We know they don't. Many of them will die in the process. And is that they are failing to protect themselves using these stop network strategies, maybe?
Yeah, I want to dig a little bit more into some of this. So let's talk about lactate a little bit more. And if I kind of put together what you said with everyday experience and what I learned when I was, in school, in grade school, really, I learned that, okay, you work out and your muscles start to burn. That has something to do with lactate and lactic acid. And we sort of used to, or I learned about this stuff as, okay, that's like a metabolic waste product. It's just sort of this passive thing, and it's like the fumes off, being given off by my cells after I'm using too much energy in the gym. But what you're basically saying is that, lactate can diffuse out of cells like our muscles. And if you think about it, like when you go to the gym and you start working out and your muscles start to burn, the fact that you have that experience means that something, in this case, like lactate, is moving around and doing stuff at the level of the muscle. But the fact that I can experience it as a burning sensation means that signal's now gotten all the way up to my brain. And you're saying this is actually actively used as a signal, not just at the muscle, but all the way in the nervous system. And it's probably to do with, how energy is being provisioned and allocated given the demands of what I'm doing, say, in the gym.
That's right. It's not only to the brain that lactate is diffusing throughout your body. And it's leaving a permanent change, permanent at least in terms of hours. So you go with your lactate to 6 millimolar, which is 6 times as much. This will be moderate exercise. Say you go for a run, and after 20 minutes run, you have 6 millimolar lactate in your blood, and that will change the mitochondria of your entire body for the next 6-7 hours. And of course, exercise does multiple things. Muscle is releasing hormones, it's releasing many things. So this would be one more in the list of ways that cells throughout the body are talking to each other. And locally, it's also similar. Even if you don't go to the gym, you start moving your legs, and lactate goes up in the whole brain. And it's locally produced by astrocytes, by glial cells. And we are very much interested in the mechanisms whereby neurons instruct these astrocytes to release the lactate. And that lactate is used as an energy substrate by neurons, but it's also modulating the future of the network by these receptors located at the surface of the neuron, G protein coupled receptors.
Yeah, I want to ask you about that too. So how is lactate in this case actually functioning as a signaling molecule? What sorts of receptors is it binding? Can you talk a little bit more about that?
Yeah, multiple mechanisms, which we think they work on different timescales. So when lactate goes in the cell, say released by astrocytes on neuronal cues. So neurons release potassium. They release nitric oxide, they release glutamate, and then lactate is produced by astrocytes and oligodendrocytes and goes into the neuron. So when lactate goes into the neuron, it's metabolized by LDH, this lactate dehydrogenase, which is the first enzyme of the root. There is a site entry to the core. And then that reaction produces NADH. And NADH is a magic molecule because the concentration, the normal concentration of NADH is almost zero. So when you have an enzyme producing NADH will change a lot the local concentration of NADH. And we know that NMDA receptors, which are located in the postsynaptic neuron, are very close to LDH, and they will get modified in their structure by this NADH. So lactate changes neuronal sensitivity to glutamate just by getting into the cell. But before that, lactate interacted with G protein coupled receptors that will signal through cyclic AMP and other pathways. And then lactate into the mitochondria will change the lactylation state, will change the activity of transcription factors. So a single part of the core, which is lactate, will change hundreds of protein activities in the target cell. And this is just one of them. There are many others being released by astrocytes in response to neuronal activity.
Yeah, yeah. I mean, I think this is a great illustration. It's starting to get at the idea that, right, the body is one integrated unit. It's not your body and your brain. It's all plugged in together. And even these core metabolites that we think of as very basic and very primitive and very simple parts of basic cell physiology, they are participating in signaling and crosstalk between muscle tissue, brain tissue, everything else. And the whole thing is really just uh, hooked up and integrated together. There's a series of metabolic reverberations that go across all of these tissue systems, and you ultimately can't separate them at some level. It's like the metabolism of your muscle is going to influence ultimately the metabolism of your neurons, and vice versa.
That's true, and tip of the iceberg as well. We can say these things about lactate because we have genetically encoded sensors for lactate, so we can see the lactate changes in real time in an animal which is walking. So, but there are about 5,000 metabolites in a typical cell, and we don't know what most of them do.
Mm-hmm.
And you have these metabolic pathways sheets in biochemistry books that this was mapped, say, 100 years ago, 100 to 50. And now that we're developing these sensors, every time we look at a new one, it's a new world. It's a new surprise. We thought metabolism was kind of done.
Mm-hmm.
Probably when you were doing your courses, you say, oh, yeah, we know metabolism and we know the glucose thing. But we really don't know. It's like having a telephone book. Of course, we have the list of people in the telephone book. What do they do? Do we know what they do by just looking at the names? We don't.
Mm-hmm.
So it's very exciting times that many labs in the world are developing these sensors, and we start looking at these things in real time in the right compartments, in the right cells. We see these exchanges, the information flowing, ebbing and flowing here and there. Beta-hydroxybutyrate is a super interesting one. The ketone body, keto diets.
Yeah. Yeah. So BHB, I've talked about this before, but for those who don't know, beta hydroxybutyrate, it's sort of the fatty acid equivalent of glucose, right? It's like what your body uses when it doesn't have glucose to do energy stuff. So if you're fasting or you're doing a ketogenic diet, BHB is one of the principal ketone bodies. So yeah, tell us a little bit more about BHB, and again, this is gonna be another example where it's doing more than just, being a substrate for energy transformation.
Yeah, sure. One of the questions, the question started when people realized, like, in exercise with lactate, when lactate goes to, say, 6 millimolar, the brain stops using glucose. So glucose consumption goes down by 30%. The same happens with the beta-hydroxybutyrate in starvation or fasting. So as blood levels go up, the brain reduces their glucose consumption by 30%. So that means that the very fact of these things being in the bloodstream, they change the brain. And now, what is going on there within the brain? We don't know so much about beta-hydroxy. We know neurons and astrocytes will change the metabolic core in a very radical way. And we know now that there are also G protein coupled receptors for beta-hydroxybutyrate. And those are neuroprotective as well. So it's like you have this parallel development of two strategies. One that comes from sugar, which is lactate, and the other from fats that will change the tissue when things go differently, either exercise or starvation, which are two ways of stress the body.
Right. So BHB, beta hydroxybutyrate, it's a ketone body. We said before that the brain primarily uses glucose for energy. The vast majority, it's sort of preferring glucose as a substrate for energy transformation. Glucose can be transported into the brain fairly readily, right? And lipids are not being transported into brain tissue as much or as quickly. If you have BHB in your bloodstream, either because you're fasting, you're doing a ketogenic diet, or even you're just taking, say, a BHB supplement, which is fairly common these days, does that BHB actually get into the brain and serve as a signaling molecule simply because it's
Yeah,
there and it's getting in?
yeah. For that, you need glucose levels to go down. So it won't happen just having ketos and ketone bodies in your diet. So you need to be more extreme, either fasting or a serious ketogenic diet in which you don't consume any glucose. It's enough a little bit of carbohydrates in your diet and that is broken.
Got it.
So this is a device that probably evolved for starvation, which was part of our life. Actually, we're used to the supermarkets now. Nobody gets
Right.
starved
Right.
in most countries, but in old times, we would go through starvation frequently.
Right. Yeah. And I can imagine, obviously, a wild animal often has no choice. If the winter's brutal, you're not going to eat for a while. You're going to go into starvation mode, not because you're choosing to fast, because you're worried about your health or you're trying to lose weight, but because there's no food around. And then I imagine over time, right, even outside of explicit starvation conditions, it's interesting that a lot of people had seasonal changes in food that, we couldn't store things like we do today and we didn't have supermarkets. So you had to go with the sort of seasonal rhythms of nature. And even though if you go back far enough, starvation was just a part of life. It's kind of interesting also that most cultures, even since, we had farming and everything, retained some kind of fasting ritual. So for all of human evolution, basically, we've had some kind of forced or chosen fasting pattern baked into life.
Oh, that is fascinating. And even in more recent times, so people wanted to get religious inspiration. They go into fasting.
Yeah.
So maybe looking for that change in the brain. What do we know from the biochemical point of view that during those periods, both exercise and starvation, you have a kind of eustress, like a low-level stress that will will increase autophagy, will increase release of the clearance of misfolded proteins in neurons. So maybe that was incorporated into culture because people felt better, felt good, and actually that prepared them for the real starvation that came later. This preconditioning is something that we do when we do exercise all the time, right?
Right.
Because we do low-level exercise, we are prepared for the high-level exercise.
Right.
That training, this preconditioning of the body, is something that is good for your neurons, good for your astrocytes, that cleans them from stuff that is not wanted.
Yeah, it makes a lot of sense. Yeah, if the first time you have to sprint is to run away from a saber-toothed tiger, you're probably not going to outrun it. But if you've been preconditioning, you can do that, and you can sort of just generalize this concept to a lot of different contexts. So, yeah, this idea that so of starvation, really, ketosis being a signal of potential starvation, you're going from glucose-based metabolism to using things like BHB more. Can you talk a little bit more about what is going on in the brain and in neural networks when these starvation indicators are coming online and you're starting to go from glucose to BHB, you're starting to go into ketosis? In terms of the signaling and how the neural networks are being reconfigured, functionally, how are they changing? How does that tie into maybe sort of the ecological needs of an animal in a starvation or pre-starvation state?
Sure. We have a few answers on the cell level. So people have thrown ketone bodies to cultures and slices and see what happens with the currents, with the cell functions. And it is very much like lactate. So you have a hyperpolarization of neurons. So they become less active. They spend less energy. And this is through activation of a certain specific type of potassium channels, the KATP channels.
They're basically becoming less excitable and therefore sending out fewer action potentials. And because even one action potential takes quite a bit of ATP, this you can immediately interpret that as sort of an economical or energy conservation mode of being.
That's right. You go into less energy mode. I don't know whether you have done a little bit of fasting yourself, but when you do that, I remember trying the intermittent diet a few years ago just for fun, and you really feel whacked. So you feel like not working at the beginning, and that correlates with this depressing effect of BHB. At the same time, the BHB effect on astrocytes is to depress glucose consumption.
Mm-hmm.
So, in other ways, the BHB pushes glucose away and becomes the main fuel for many cells. Never 100%, but to a certain extent, which would be 30, 40%. So, BHB displaces glucose, so the cell will use BHB now because it's a nice fuel. And you burn the BHBs to CO2 as you did with glucose before, and the cell can use glucose for purposes which are more strategic. Say, in the case of neurons, glucose is not just used for energy. Glucose is very important for protection against reactive oxygen species. So one of the first pathways when glucose enters glycolysis is that it's directed to the side pathway, which is called the pentose pathway. And the pentose pathway produces NADPH, which is the main antioxidant of the cell. So you will not only produce more energy, but boost your antioxidant defenses just for BHB being present.
Interesting. I want to talk too about, thinking about how metabolism and information processing are inextricably linked, both at the cell level, but all the way up to, so at the level of cellular energy metabolism of one cell, all the way up to the systems and organismal level. I want to thinking about ATP, adenosine, and just the behavioral state of the animal, being awake versus being asleep, and sort of how all of these things are integrated. So again, if we think about the simplistic way of segregating metabolism from information processing, we think of ATP as the energy currency of the cell. That's part of this metabolic core. And, immediately when you start to look at what's going on in the nervous system, you realize, there's a lot more to it than that. So even ATP itself is released by neurons, almost like a neurotransmitter would be released. Can you start talking about the role of ATP in intercellular signaling in the nervous system, especially in the context of how this starts to relate to things like sleep-wake patterns in the whole organism?
Right. This is the third, I would say, angle of this core metabolism talking to the network.
Mm-hmm.
So what happens during the day? ATP is normally millimolar concentrations within the cell, and this is used for all the wonderful things the cell is doing.
Mm-hmm.
But during wake, a little bit of ATP is being degraded into adenosine.
Mm-hmm.
So, and this happens in direct relationship to neuronal activity. So as you transit throughout the day, your adenosine levels in the extracellular space, they go slowly up and up and up and up.
Mm-hmm. I'm using more ATP, I'm doing more stuff. And as a natural consequence of that, adenosine is being produced and sort of slowly building throughout the day as a function of how long I've been awake and how energy intensive my behavior has been.
Exactly. Adenosine is the degradation product of ATP. ATP is made of an adenosine molecule and some phosphates.
Mm-hmm.
So adenosine is produced by this normal process of activity. And you have this accumulation in certain areas of the brain, the hypothalamus, some nucleus of the brainstem. And this is understood as the main trigger of sleep. So At the end of the day, your adenosine levels are higher and sleep is induced. You have a charge of sleep charge, so to speak. And then during the night, your energy consumption in the brain in general goes down by about 20 or 30%. So you recover, adenosine is recycled into ATP, and you recover your level and you are happy and fresh in the morning. So sleeping is important for many reasons, metabolically too. So this cycle of adenosine is very well in register with the cycle of sleep-wake. Something interesting is that this is not only active, this adenosine signaling throughout in that, digamos, say, long time courses. Adenosine can change very quickly when you activate a region. So if you are using your hand, adenosine levels around the motor neurons that are moving the hand will go up. And this is understood as well, again, through G-protein coupled receptors as a way, as a mechanism of, to say to the network, be careful, stop too much activity. So adenosine is in short-term times is used as lactate and as beta-hydroxybutyrate as a signal of mind. Be careful with the activity, and that happens in every single part of the brain in relation to the local activity of the network.
Yeah, so there's multiple there's multiple scales of spatial and temporal regulation here. It's not just that adenosine is this slow thing accumulating everywhere evenly as just a global signal. It is also localized. If I'm using my left arm a lot, the motor neurons controlling that part of my body will be getting a different level and time course of adenosine-based signaling than other parts of my brain.
That's true.
Yeah. So interesting.
And these three signals are also used for something we call lateral inhibition. So if you have a region of the brain that is using a lot of energy because it's being activated by the network, like the motor neurons of that moving your hand, this product that diffuse away from that region will inhibit regions which are very close by. So that has consequences in terms of quality of the action. So when you move your hand, you don't move your shoulder at the same time because those are being inhibited. And also in terms of metabolic recruitment, something we call this term is used for the harvesting of resources by the active area, active zone. So this diffusing core metabolites signal, please not just don't stimulate me too much, but also give me resources, you lazy guys, which are doing nothing at this time.
So you say that, so adenosine has a neuroprotective role, kind of like lactate does in the context of exercise, BHB in the context of fasting. So adenosine is sort of related to how long have I been awake, how much have the cells been doing something. Lactate is very much tied to exercise, right? Like I'm working out my muscles, they're starting to burn. Lactate is a signal behind that. And then BHB is sort of a comparable signal in the context of fasting or starvation. We're running out of energy inputs. And so BHB goes up and tells the different networks in the body that that's the case. Why are things like lactate, BHB, and adenosine neuroprotective? What's sort of the common denominator here, and why was this a way that sort of evolution seemed to set things up, where you have these core metabolites that serve this protective role, in this case for neurons, when we are under stress-like states like exercise or fasting or just prolonged wakefulness?
These guys come on that they are at the interface of big regions of the metabolic network. So glycolysis is the first, I would say, domain of the metabolic network that comes from glucose to lactate, let's say. And then lactate or pyruvate, which is the form of lactate that goes into the mitochondria, gets oxidated. So you have these two ancient regions of metabolism that come actually from different bugs. We think that mitochondria, you probably had a podcast about this one before. So mitochondria and glycolysis, they have different genetic origins, different bugs that got together and did produce eukaryotic cells. So, and lactate is in the interface between the two. And now beta-hydroxybutyrate is in the interface between mitochondrial oxidative metabolism and lipid metabolism, which is in the cytosol as well.
So lactate's telling the body that about the balance of oxidative phosphorylation from mitochondria versus glycolysis, two different modes of energy production that are each have a different level of efficiency. BHB is at the interface within the mitochondria between fatty acid versus glucose-based metabolism. And okay, so basically what you're getting at here is this: they sit at the interface. These are molecules positioned perfectly at the interface of two different modes of energy production. And therefore, when one of them goes up or one goes down, we start using one mode of production more than the other because we're running out of energy because we've been exercising a lot, say. These molecules go up and they serve that the place they're positioned at this interface between one mode and another mode of energy production makes them very well suited to serve as this kind of signal to the rest of the body, the rest of the cells and tissues, that something is changing. We need to start behaving differently and reallocating our resources.
Yes, and it's not just the location, it's that they are diffusible. So both of them, lactate and BHB, they are moved by monocarboxylate transporters, which are very abundant on the surface of most cells. So is where they are, and they can move in and out. So, for instance, in diabetic people, when they go into a crisis, they transform most of their fat into ketone bodies, and you have a ketoacidosis. And this is this ancient device being going hayward. So, and now a more precise phenomenon for us neurobiologists is that we suspect that beta-hydroxybutyrate is also produced in the brain. It's something we don't know for sure yet. But lactate it's not just comes from exercising the circulation, and beta-hydroxy from starvation in the circulation, but we think now that this can be also very local, so that glial cells can produce lactate and BHB. Lactate, we are pretty sure about that, but BHB this is new research. We suspect from data in Drosophila that you could have this circuit local as well, that will signal local mismatches between fats and glucose and mitochondria. These domains of metabolism will inform the tissue from what's going on metabolically very locally.
And so this gets us to this concept of scale spanning coordination that you've written about. So explain that for us and how it ties into what you're talking about here. We have to have multiple spatial and temporal scales at which cells are talking to each other to coordinate changes that are ultimately rooted in how much energy is available, what type of energy is available, and how it should be partitioned and allocated amongst all of the cells and tissues.
Thank you for that question. This is the title of the paper, actually, which is an idea that is not totally finished. We are working on it, but here's the wrap. So we think about neural networks, and this is very intuitive, right? Big ensembles of neurons spanning 10 centimeters in a human brain. And now we talk about metabolic networks. The very big difference between neural networks and metabolic network is their size. So neural networks are typically from, say, 100 microns to 10 centimeters. That's the scale. They span 2 or 3 orders of magnitude. And metabolic networks, they live in a different part of the scale, of the hierarchy. So the biggest possible metabolic network is about 100 microns. Why is that? it's because of diffusion limitations.
Yeah, some of these molecules, the ones that we've mentioned that are part of the core metabolism of cells, they can diffuse, and that makes them really useful as sort of unbiased signals about energy metabolism. But diffusion is slow, and you can only diffuse so far, so fast. So they're limited in spatial scale.
That's correct. So if you look at the concentration of, say, ATP, it's about 1 millimolar in the tissue. And now we know the speed at which ATP is being used. Then you can calculate something which is called the turnover time. So how long does it take to renew itself? And for ATP, it's about 1 second. In the order of 1 second to 6 seconds, depending on let's say 10 seconds to be optimistic. So you need to solve the ATP problem in 10 seconds. We know that, practically, because if you run out of oxygen in 10 seconds, you are unconscious. Mm-hmm. So oxygen is about 1 second, the turnover time. Pyruvate is about 30 seconds. So these things, they move very fast considering their concentration. And now, if you take that time and you see how fast they diffuse in that time, you realize that everything that is to do with ATP has to happen within 50 micrometers. So you cannot, if you're using a lot of ATP, the ATP that is a millimeter away will not help you.
Right.
So that puts a cap to the metabolic unit of the brain tissue, which is about 200 microns. Whatever you have to do within those 200 microns, or that won't work, which makes a lot of sense in terms of the intercapillary distance, which is about 50 microns in a human. So now we have the whole metabolic thing that neurobiologists like me have been working for years has to be solved within 200 microns. But the network, the signaling network, is centimeters. So how do these two networks talk to each other? And this is where these metabolites come along. So when lactate is released from a region, it diffuses away and can reach 100, 300, 400 microns and tell the network something that the network wouldn't know otherwise. So there's no other way the network can know that your resources are being stretched other than these mechanisms based on diffusible core metabolites.
Interesting. Yeah. So yeah, there's a spatial and a temporal component to this. You can't, if a cell runs out of energy, it takes a certain amount of time to replenish it itself. And so it has to, and because all of the cells are sort of hooked up to other cells at some level, and they all depend on each other at some level, there has to be a way for cells to communicate their energy needs and their energy supply and the current demand of things within a given time window and at a given spatial scale. And all these things are constrained in very important ways.
Yeah, you can draw a metaphor in terms of our own lives. You could say that, you have your program, your podcast is fine, and you have certain requirements by the world. You need to pay your taxes. All that is okay. This is the network, the human network, telling you what to do, when to do it. But then you have to eat, right? You have to breathe to keep your own integrity as a part of the network. So how do you say to the whole world, please, I'm working too much or I'm working too little? You need ways to transfer that information. And we normally haven't thought that way because we took for granted that the pieces were designed for the machinery. So that the brain was okay, the brain works, evolutionary design, no problem. I will not think how it gets to do the job. This is a top-down way to think about the problem. If you think bottom-up, you say, look, we have the poor little neuron, the astrocyte, which is very close to the neuron. How this unit, this neurogliovascular unit, survives for 80 years? How does it keep the the network at bay.
Mm-hmm.
And this is probably one of many mechanisms that act at different timescales. But we're very excited because we're starting to look for more different spatial-temporal domains and start looking and finding more ways in which this spanning is achieved. It's a communication between domains that otherwise wouldn't talk to each other.
Mm-hmm. I want to talk a little bit more about the fact that the brain is very energy intensive. So there's a bajillion neurons in our brain, and they're firing off action potentials all the time. We're dealing with really big numbers here. Exactly how expensive? Can you give us more of a concrete sense of how expensive neural firing is? How expensive in terms of ATP are action potentials? I want to talk about that, and then I want to talk a little bit more about some of the work you've done around glial cells and how they integrate into the picture from an metabolic energy and metabolism perspective, and how they're doing more than just sort of being passive supporters of what the neurons are doing. But let's start with the neural energy demands of the brain. How big are they exactly, and how expensive are things like action potentials, the signals that underlie our cognition and our behavior and all the things that brains are doing?
It is the issue of the brain. So a way to put it is that on average, uses energy 10 times faster than the rest of the body.
Mm-hmm.
If you put this energy consumption in muscle, would be like running a marathon all the time. Now, how is this spent? About 70-80% of the consumption is to do with the signaling itself. So sodium ions get into the cells during synaptic potentials and action potentials, and this is in itself free, but you have to send them out later to recover the balance. And this is done by sodium pumps and calcium pumps. And this is where most of the energy of the brain is used. We knew that from old, very nice experiments in which people would just block the sodium pump and energy consumption by the brain would drop by 60%. If you're sleeping, it doesn't change very much because the brain is very active during sleep anyway. But goes down by 30-20% in some areas. So this is how the energy is spent. Sorry, I got distracted. What was the other part of the question?
Oh, so a lot of the energy is spent, and a lot of the reason that brains and neurons are expensive is because the signals they send, like action potentials, depend on constantly maintaining and remaking gradients of ions. And gradients are expensive because from the basic physics perspective, right, the gradients want to run down. Things want to go from high to low. Things want to go to equilibrium. So you have to constantly literally pump stuff in and out of the neuron to maintain these gradients. But then the gradients sort of dissipate or go down as the neuron sends signals. So it literally has to do a lot of work to physically pump stuff this way or that way to maintain gradients. And if that didn't happen and we didn't have the energy currency of ATP to make it happen and do that work, then our brains would stop working.
That's correct. So the sodium pump, the calcium pump are the guys, they're very slow actually. So you need millions of them. So for every channel that allows sodium in, which sodium channels are very fast, you need about 1 million pumps because they pump at rates of, say, 20 per second. And an ion channel can allow 1 million per second. So little channel doing the clever job and the sodium pump doing the slave for minutes after this has happened. And this multiplied by so many synapses gives you this super rate of glucose and oxygen consumption. And now our brains are bigger than the average brain of a mammal.
Mm-hmm.
So this is multiplied by a kilogram of stuff. So then comes the hypothesis of the expansion tissue hypothesis. I don't know whether you heard about that one.
Yes, I
But
have.
which is quite interesting. I'm not sure that's been demonstrated to a certain extent, but the idea that
Yeah. What's the idea here?
here is that we evolved our primates, our big brain, in terms of food scarcity. So how did we do that? Nowadays, it's no problem. We have supermarkets, right? But during evolution, we were starving most of the time. We know that because of biochemical and gene markers of those hardships. And the idea is that we got rid of some energy expenditure in other organs, mainly the intestine.
Mm-hmm.
So when we became carnivorous, we can use more time for looking for food, and we don't need such a long intestine.
Mm-hmm. So conceptually, the idea here is if you just look at the human body compared to other primates and other animals, we've got less of a GI tract. There's less stuff down there, and our brains are bigger, relatively speaking. So the question is, well, how do you power that energetically? Well, if you start eating foods in ways that allow you to more efficiently extract more energy, then you can power that bigger brain and not have to have as much of a GI tract to pre-process it. So, for example, if we start eating more animal products that have a higher density of bioavailable nutrients that don't require as much pre-processing in the gut, if we start cooking our food, essentially pre-digesting it, we can extract that energy faster. And the idea is we sort of took some of the GI tract and the investment in that other animals have, and we could put it towards the brain because we started getting the fuel in a more efficient manner.
Yeah, it's very seducing theory. I don't know how to demonstrate. Well, it's difficult to, but it makes kind of sense to me. And now we have felines, which are carnivores, and they don't have big brains. So yeah, the discussion goes on. But maybe
Right.
the brains wouldn't be so big otherwise. So it's tricky to try and understand that. But what we do know is that our brain is using 20% of our energy intake all the time, even if you're sleeping. And that 20% is a big number.
Yeah. And so talk a little bit more about glial cells, things like astrocytes. What are these other cell types in the brain that are not neurons? And how is the picture of these things, how has that started to change over time into the present moment? And how does that tie into some of the fundamentals of what we discussed around core metabolites and intracellular signaling?
Yeah, that is a long story. You know, these cells were discovered almost at the same time as neurons. They were called glial cells because Rudolf Virchow thought they would be the glue between neurons, keeping things in place. So from the beginning, they were kind of demoted to a structural role. But nowadays, I think we know it's much more interesting than that. They're not just a way to outsource a lot of the metabolic pathways of the neurons. So neurons do wonderful things. They're very super fast, but metabolically they are quite primitive compared to glial cells. Glial cells, astrocytes, oligodendrocytes are super sophisticated in terms of pathways, and many of the substrates that neurons use day by day are produced by the local glial cells. So they have this nourishment aspect, but also signaling. So when glutamate is released and does the thing, and you get neurotransmission, that glutamate is taken by glial cells, not by neurons. And that puts a loop into the cell that is not a neuron that will control the glutamate cycle. And there are many parallel examples of glial mingling with the neuronal function. Recently, about 20 years ago, people discovered that now glial cells release neurotransmitters. This is called glial transmission. So they're not just nourishing, modulating, they can change the circuit directly by releasing neurotransmitters. And most diseases today, they are thought to have a glial component. So mutations in proteins which are neuronal, that are known to produce a Alzheimer's or Huntington, whatever. If you just mutate the neuronal protein, you don't get the same disease as you also mutate the glial protein. So somehow glial are participated in the node of information processing, and also in terms of nutrition, control, etc., etc. Every astrocyte in gray matter controls about 10,000 synapses. And what happens in those synapses just not depends only on neurons, but in that particular astrocytes and in the inputs he's receiving from all parts of the brain, many different neurotransmitters. So people trying to simulate networks, now they are thinking about putting glial cell similar stuff to account for that modulation. They're much more sophisticated in terms of the signal. Neurons are quite digital. You know, they fire or they don't. Even though they receive synaptic input and they kind of integrate this input into an action potential. Once the action potential flies, it's going to be it or not. But glial cells are much more nuanced. They release different substances. They release the lactate we were talking about before comes from the glial cells, not from the neurons. And probably the beta-hydroxybutyrate. So it's kind of a new area in neurobiology, the glial biology, and which is sprouting. Glial meetings used to have 100 people 20 years ago, and now they have 10,000. But neuro meetings, they used to have 30,000, and now they have 20,000. So people are looking at glial cells and say, oh, I think we need to know a little bit more about this guy, which we know much less about.
Yeah. What are some of the questions and the projects that you're working on in your lab today that you're really excited about that are related to energy metabolism in nervous systems?
Well, the key word would be mitochondria. In the simplified way to look at things from 20 years ago, we thought the interesting cells, the neuron, has lots of mitochondria and just all the energy the ATP is producing mitochondria and so on. Now we know glial cells, astrocytes in particular, they have as many mitochondria as neurons, but apparently they do differently. And now they have domains, they have processes in which mitochondria look very different morphologically, and the same for neurons. So you have mitochondria in dendrites, mitochondria in somata, mitochondria in axons, they look very differently. And with these new techniques that have been developed in recent years, we can now look at the behavior of single mitochondria. And this is a new it's like having a telescope that you looked into dark and now you see galaxies, right?
Right. It's like literally you need a microscope to see into the brain to see the neurons and the glial cells, but then you need a different type of microscope or a bigger zoom lens to see the networks of mitochondria within these cells. And each neuron, each cell is really a network of other mini cells in essence.
Yeah. So you choose your scale and you can get lost forever in your scale. So we love very much communication between cells, but now the communication around the synapse, the mitochondria that are there telling things to the next synapse or not to the next, that will depend on phenomena which are much faster and smaller. What we can do is we can target now sensors for these metabolites and others to single mitochondria and look at that. And
we are in single mitochondria within a neuron or some other cell.
Yep.
Wow.
Yeah, and imagine any neuron ordinary small neuron in your cortex, they have 1,000 mitochondria. And now we know some of the mitochondria are bound to the plasma membrane, some are close to the nucleus, some are close to the ER, endoplasmic reticulum. Some of them are very close to a synapse, to the dendritic spine or to the button. So there is a new complexity that is surfacing because now we can see things that we couldn't before.
Yeah. Wow. Are there any final thoughts you want to leave people with, or anything you want to reiterate from our discussion with respect to the interface between metabolism and information processing within the nervous system?
Maybe final thought is that we are really beginning. Some people maybe can think that we know a lot about the brain. I will give you one example. We have these sensors for about 25 metabolites, but there are 5,000.
Right.
And we don't know what they're doing.
So we can't even watch 1% of these metabolites.
No. And just to give you an arcane, very obscure metabolite that wasn't even in the interest of anybody about a year ago, kynurenine. Kynurenine is even difficult to pronounce. So this is something that people interested in metabolism like me would avoid, kynurenine. What do we know about kynurenine today? This is thanks to work done in Stanford in Alzheimer's mice, that very early during disease development in animal models of Alzheimer's, you have a change in kynurenine metabolism in astrocytes. And if you inhibit that change with a drug that is available for cancer treatment, those animals don't develop Alzheimer's. And this is one of the 5,000 that we mentioned before that we cannot map. So, but we know how to produce sensors for all of those. In principle, in say 20 years, we will have maps for hundreds of them, not just the 25 that we can see today. So the future is very nice for people that want to look at metabolism. Don't be scared of the word metabolism. It's scary for many people that like neurosciences, because now we can see things with microscopes and they look beautiful. All these sensors are fluorescent and we can see them working in real time. So it's a new era. I'm very optimistic about the future of this field.
Yeah. When we finish here, would you be willing to send maybe some short videos that we can share with people so they can literally see what you're talking about?
Of course.
Yeah.
I will gather those for you. No problem.
Great. Well, thank you very much for your time. This is fascinating stuff. I think this is a very fascinating area within neuroscience and just within biology generally. And as you said, we can't even look at even 1% of some of this stuff yet because we don't have the tools available. So I just want people to appreciate that even though there's all of these studies and all of these things we've learned for centuries, we haven't even understood the tip of the iceberg when it comes to biology. And I think that's a very humbling thing that people should keep in mind when we start to get a little bit overconfident in how much we think we know about how all of this stuff works.
Well said. Thank you very much.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.